double-stranded break repair, and homologous recombination. In this chapter, we will examine the molecular processes of recombination in prokaryotic and eukaryotic systems. We'll begin by exploring the enzymes and mechanisms involved in prokaryotic recombination and how they compare to eukaryotic repair strategies. We'll then differentiate between mitotic and meiotic recombination, both of which play critical roles in maintaining genome integrity and generating genetic diversity. Finally, we will analyze how recombination is harnessed as a tool in genetic engineering to create gene knockouts for research and therapeutic applications. Recombination refers to the exchange or swapping of genetic material between DNA molecules, typically between homologous chromosomes. This process occurs naturally as a method of genetic repair or diversification. In prokaryotes, recombination happens through mechanisms like conjugation, transfection, and sometimes transduction. In eukaryotes, recombination is a hallmark of meiosis, where crossing over events occur. Importantly, recombination is also an essential laboratory technique used to generate recombinant DNA molecules, which form the basis of many genetic engineering strategies. Double-strand breaks are among the most severe forms of DNA damage because they remove the complementary sequence needed for repair. To manage this challenge, cells utilize two primary repair pathways: homologous recombination and non-homologous end joining. Homologous recombination uses a homologous DNA sequence as a template for accurate repair, while non-homologous end joining directly ligates the broken ends together, often with some loss of information. Both pathways are essential for genome stability, especially in response to DNA damage or replication stress. In bacteria, the collapse of a replication fork is a critical event that halts DNA synthesis. To restart replication, the cell initiates homologous recombination. The process begins with the formation of a single-stranded 3-hydroxyl tail at the site of the break. This tail invades an undamaged homologous sequence, forming a structure known as a Holliday junction. As the Holliday junction migrates, it facilitates the repair and restart of the replication fork. Resolution of the junction and subsequent ligation restore the replication machinery to its functional state. The molecular choreography of replication restart in bacteria involves several key proteins. RecBCD binds to the DNA at the break site, processes the blunt ends, and generates a 3' overhang. RecA then binds to this overhang and promotes strand invasion into the homologous DNA duplex. RuvC facilitates branch migration of the resulting Holliday junction, while RuvC cleaves and resolves the junction. Finally, PriA reestablishes the replication machinery, allowing synthesis to resume from the repaired fork. This slide provides visual reinforcement of the RecBCD complex and its role in processing double-strand break ends. The figure highlights the sequential actions of DNA binding, helicase activity, and nuclease functions that produce the 3 overhangs necessary for homologous recombination. These illustrations help contextualize the mechanistic steps outlined in the previous slides. In eukaryotic cells, mitotic recombination primarily serves as a repair mechanism for double-strand breaks during the late S or G2 phases of the cell cycle. During these stages, a sister chromatid is available to provide an accurate template for repair. The cell uses one of two homologous recombination pathways: synthesis-dependent strand annealing or double-strand break repair. Both pathways begin with similar early steps, including resection of the break and strand invasion, but diverge in how they complete the repair. The first step in mitotic homologous recombination involves the processing of the double-strand break to generate 3 single-stranded DNA tails. Exonuclease digest the DNA ends, exposing these single-stranded overhangs. These tails are essential for locating and pairing with homologous sequences on the sister chromatid, which will serve as the template for repair. Once the single-stranded tails are exposed, they are immediately coated by replication protein A, or RPA. This binding prevents secondary structures from forming and signals the cell to pause the cell cycle. Rad51 then displaces RPA and forms a nucleoprotein filament capable of locating and invading the homologous sequence. The tumor suppressor BRCA2 plays a vital role in helping Rad51 load onto the DNA. Mutations in BRCA2 are associated with increased cancer risk due to impaired homologous recombination. In the synthesis-dependent strand annealing pathway, the invading single-stranded DNA tail is extended using the homologous template. After synthesis, the newly formed strand is displaced from the donor duplex and reanneals to the complementary sequence on the original broken molecule. The remaining gaps are filled and nicks are sealed. This pathway restores the original sequence without generating crossovers, making it a conservative and safe repair strategy. In contrast to synthesis-dependent strand annealing, the double-strand break repair pathway involves capturing both ends of the broken DNA. The invading strand is extended, and the second end of the break is also paired with the donor template, forming a double Holliday junction. This structure is then resolved, potentially leading to crossover events. Although this pathway is more complex, it ensures robust repair of severe DNA damage and can result in genetic exchange between chromatids. When a homologous template is not available, such as during the G1 or early S phase, cells rely on non-homologous end joining to repair double-strand breaks. The Q protein quickly binds to the broken DNA ends and recruits the DNA PK complex. If the ends are compatible or clean, a specialized ligase can directly rejoin them. However, if the ends are dirty or incompatible, limited processing is required, often resulting in mutations. This mechanism is essential for processes like immune receptor rearrangement, but carries a risk of introducing genomic errors. Gene conversion is a process that occurs during recombination where the genetic information from one DNA sequence is non-reciprocally transferred to another. In the context of homologous chromosomes with two different alleles, gene conversion can change one allele to match the other. This phenomenon plays a significant role in genetic diversity and, in some cases, can disrupt normal gene expression by rendering one allele non-functional. During meiosis, homologous recombination becomes a central feature of gamete formation. It promotes genetic diversity by reshuffling alleles between homologous chromosomes. Gene conversion can also occur during this process, altering segregation patterns in gametes. Meiotic recombination ensures that each resulting gamete contains a unique combination of parental alleles, which contributes to the variability observed among siblings. Meiotic recombination is more frequent and specialized compared to mitotic recombination. This is due in part to the activity of SPO11, a meiosis-specific endonuclease that creates intentional double-strand breaks across the genome. These breaks are necessary to ensure that each homologous chromosome pair undergoes at least one crossover event. During meiosis I, the preferred repair template is the homologous chromosome rather than the sister chromatid. The recombination process is facilitated by two recombinases, Rad51 and DMC1, both members of the RecA protein family, with DMC1 being uniquely expressed during meiosis. Meiotic recombination relies on a coordinated set of enzymes that generate and process double-strand breaks. The SPO11 protein initiates recombination by creating the double-strand breaks required for crossover formation. Once SPO11 is covalently linked to the DNA ends, the mRNA complex, along with CDIP, removes SPO11 and begins processing the break. Further resection is carried out by exonucleases like EXO1 or the DNA2 helicase complex to generate single-stranded 3' overhangs. Strand invasion is then promoted by Rad51 and the meiosis-specific recombinase DMC1, which facilitate pairing with the homologous chromosome to initiate either synthesis, dependent strand annealing, or full double-strand break repair. In this pathway, the 3' single-stranded tail invades the homologous chromosome and primes new DNA synthesis. After synthesis, the extended strand dissociates and anneals back to the other end of the original break site. This results in the formation of a heteroduplex that may contain mismatches. These mismatches can be repaired in a way that causes gene conversion, effectively replacing one allele with the sequence from the homologous chromosome. Importantly, this mechanism does not produce crossover events, making it a conservative form of recombination during meiosis. In the double-strand break repair pathway, both ends of the broken DNA are engaged in homologous recombination. After strand invasion and DNA synthesis, a second end capture occurs, forming a double Holliday junction. This structure can then be resolved in a way that leads to crossover or non-crossover products. Crossovers are critical during meiosis because they physically link homologous chromosomes, ensuring their proper alignment and segregation during the first meiotic division. These events not only maintain genome integrity, but also promote genetic diversity in the next generation. https:// This slide provides a visual summary of the meiotic recombination process. The formation and resolution of Holliday junctions, along with mismatch repair within heteroduplex DNA, explain how gene conversion and crossovers occur. These events are essential for accurate chromosomal segregation and for generating the genetic variation that underpins evolutionary adaptation and individual uniqueness among.
The first molecular model of the genetic exchange that occurs between homologous chromosomes during recombination was proposed by Robin Holliday. The Holliday model involves several steps. First, the homologous chromosomes are both nicked at identical locations. Then, the strands from one side of the nicks invade the homologous chromosomes, base pairing with complementary strands. The invading strands are next covalently linked to the original strands at the nick site, forming what is called a Holliday junction. The Holliday junction migrates away from the original nick site, a process called branch migration. As it does so, DNA strands are swapped between the chromosomes. This creates heteroduplex regions on both chromosomes, where minor base sequence differences between homologous chromosomes results in a region of DNA with a low percentage of mismatched base pairs. The length of branch migration may vary, but at some point breaks are made in the DNA that end migration and resolve the entangled DNA strands into two separate chromosomes. There are two ways that breaks can be made. One results in non-recombinant chromosomes, while the other results in recombinant chromosomes. If the crossed strands are cleaved by an endonuclease, then after ligation within the chromosomes, there will be two non-recombinant chromosomes with short heteroduplex regions. Alternatively, if one rotates one DNA helix 180 degrees, a process called isomerization, we can visualize how uncrossed strands can be broken. After the uncrossed strands are cleaved by an endonuclease, ligation can produce recombinant chromosomes with short heteroduplex regions.
Although gene conversion primarily occurs during meiosis, it can also happen in mitosis. In mitotic cells, the preferred donor for repair is the sister chromatid rather than the homologous chromosome. While rare, gene conversion in mitosis can have serious consequences. If an individual carries one mutated and one functional copy of a tumor suppressor gene, gene conversion during mitosis could replace the functional allele with the mutated version. This loss of heterozygosity eliminates the cell's backup copy and increases the risk of cancer, helping to explain how inherited predispositions can lead to disease. This slide presents a conceptual question to reinforce the clinical significance of gene conversion. Individuals who inherit one defective copy of the BRCA gene are at risk of acquiring a second mutation or undergoing gene conversion that eliminates the remaining functional allele. Once both BRCA alleles are non-functional, the cell's ability to perform homologous recombination is severely impaired. This increases genomic instability and significantly elevates the risk for developing breast and ovarian cancer. Therefore, gene conversion is a key mechanism underlying cancer susceptibility in BRCA mutation carriers. Gene knockout technology takes advantage of homologous recombination to disrupt specific genes in mice. This approach was made possible by advances in culturing embryonic stem cells, which can be genetically modified in vitro and then introduced into early mouse embryos. DNA constructs designed with regions homologous to the target gene can integrate precisely at that locus through homologous recombination. Successful knockout cells can be selected and propagated, ultimately giving rise to offspring in which the gene of interest is inactivated throughout the organism. The process of creating a gene knockout often involves positive-negative selection. The gene of interest is interrupted with a New cassette, which provides resistance to the antibiotic G418. Additionally, the construct contains the HSVTK gene, a negative selection marker. If homologous recombination occurs correctly, only the NEOR gene is integrated, and the HSVTK gene is excluded. However, random integration results in both NEOR and HSVTK being inserted into the genome. By treating cells with G418 and ganciclovir, researchers can select for those that underwent targeted recombination and eliminate those with random insertions. The chapter concludes with an introduction to CRISPR, a revolutionary gene editing technology. Unlike traditional knockout methods that rely on homologous recombination, CRISPR allows for precise genome editing using a guide RNA and the Cas9 nuclease. This system can be programmed to target virtually any DNA sequence, where it creates a double-strand break. The cell's natural repair mechanisms then introduce insertions, deletions, or even specific gene edits through homologous recombination or non-homologous end joining. CRISPR has transformed molecular biology and is now widely used in research, biotechnology, and clinical applications.